Is the cure for cancer in our blood? Cellular immunotherapy is quickly emerging as a cutting-edge cancer therapy, leveraging cells as living medicines to treat cancer. Chimeric antigen receptor T-cell therapy, or CAR-T, is the forerunner in the cell therapy space. But what is CAR-T? CAR-T therapy exemplifies the potential of personalized medicine, using genetically reprogrammed T-cells to precisely target cancer cells. With CAR-T therapy, T-cells are collected either from a patient, autologous, or donor, allogeneic, and then undergo transduction of CAR-encoding genes programmed to recognize a patient's cancer. After vigorous testing, the CAR-T cells are expanded and infused back into the patient with a new ability to target specific cancer cells.
The cell therapy field is really just one of, I think, the incredible triumphs of immunology and applying basic immunology to the clinic. I remember being in a seminar where Jim Allison, a Nobel Prize winner, presented some of the first patients who had been treated for melanoma with the drug that would become Yervoy. And you could just see the amazement of everyone in that room to see, the things that they had done in mice, that they had done in cell culture, really get applied to patients. And I think that was the first time that I really understood that this basic field that I was just scientifically interested in had the potential to really impact people's health. One of the most important mechanisms of action of CAR-T drugs is that they can kill tumor cells directly in the patient. And so to measure the potency of a CAR-T cell, it's really important to be able to measure their ability to kill a target cell in vitro. When you're developing a potency assay for CAR-T cells, you have to make a choice about what functions you're going to measure. And originally, a lot of developers chose cytokine production because we have great tools for measuring cytokine production. But a more direct measure of the potency of a CAR-T cell and its efficacy against a tumor is target cell killing. And that's why we developed the HiBiT target cell killing platform. So this assay is based on a split luciferase technology where the detection reagent contains a larger component of the luciferase called large bit, and the target cells have been engineered to express a HiBiT protein tag. So when the target cells are alive, that HiBiT tag is sequestered inside and is inaccessible to the large bit in the detection reagent. But when a CAR-T cell comes along and kills that target cell, it releases the HiBiT into the media, and it can complement the large bit, producing light in proportion to the number of target cells killed.